Well, I think we probably can start now. So it's my very most pleasure to introduce today our speaker, Professor Ronald de Vries, who is really well-known, international renowned specialist in fungal plant biomass degrading enzymes and on carbon metabolism in filamentous fungi. So, Professor de Vries, you have numerous international collaborations, notably also with the KZ database team in Marseille and with the JGI as a member of the Fungal and Algal Program User Advisory Committee. And you did your PhD at the Wageningen University in the Netherlands, focusing on plant polysaccharide degrading enzymes. And then first postdoc at the at the Institut Pasteur, I believe, in Paris. That was your first postdoc, yeah. And then a second postdoc also in Wageningen University. So the first one you focused on osmotic stress, if I'm correct, and the second one on colony differentiation in fungi. And then you started your own group with the prestigious research scheme from the Dutch Research Council, known as NWO. And so you had a program that's called Veni Vidi Vici, and you got VD, a starting program for your own group. And later on, you confirmed and reinforced your group with a Vici grant. And then didn't stop. You had many and numerous funding and programs with Europe and so on. So that's the formal introduction. The more personal introduction is that you hosted me for my first postdoc experience, and I have a wonderful memory of the years that I spent in your group in the Netherlands. And so that's my most pleasure to give you the microphone or the computer today for speaking on your research. Now, I want to add before you begin that your research is at the Westerdijk Institute in Utrecht, but you are going to move and take your team to the Luxembourg, right? And so you can maybe tell us more about it during this seminar.
Okay, thank you very much, Isabel, for the nice introduction. Let me just start by sharing the screen so that we make sure that everybody can see. So I hope the screen is visible to all. So today I'm probably going to go a little bit out of most of your comfort zone because I will talk a little bit about pathogens and evolution of pathogens at the end. But I'm mostly going to tell you about fungal diversity and diversity in the context of how fungi acquire carbon in natural situations and how we try to visualize that in lab studies. Now, of course, we have to keep in mind that the lab is no substitute for nature. We cannot reproduce the complexity of things that happen in nature, but what we can try to do is dissect the complexity to individual aspects and try to understand the individual aspects and bring it back to a whole system. So I'm going to try and go through that. I might overload you a little bit with data during this seminar. So please don't hesitate to ask questions. If at some point you get completely lost, you don't have to wait till the end. You just raise your hand and hopefully I will see it. And then I can always pause a little bit. So, as Isabel said, so I'm currently at the Westerdijk Institute, where biodiversity is a very important aspect. So the focus of the last 15, 16 years has been very much on comparison of fungi, but also at the same point on sort of deeper studies with some what we call reference species, and particularly Aspergillus niger I will talk about, because ultimately it's not really possible to go deep when you also go wide. So you always have to make a choice. And many of our studies, we go deep first in reference species, and then we use that knowledge to go wide, typically with omics type studies, to see if we can see the differences and make sense of it. Right, let's see. So, as I said, I'm going to talk to you about carbon utilization from natural sources, and actually from most fungi, and that's more or less independent whether they're plant pathogens or whether they're saprobes or whether they're symbionts. For many of them, plant biomass is the preferred carbon source. And plant biomass is a rather complicated carbon source because it doesn't consist of easily consumable sugars. That's also why, for instance, certain model fungi like Saccharomyces cerevisiae typically don't grow on this because they lack the ability to degrade it. So here in the picture, you see a schematic thing. Let me actually see if I can get my pointer. What? Yes. So here you see a schematic picture of plant biomass starting from a piece of wood, which typically consists of all these cells, and then actually the majority of the biomass, so the dry mass in plants, is the cell wall. And the cell wall is quite complicated. You see here these long cables of cellulose microfibrils, which are embedded with hemicellulose and then also with lignin around it, depending a little bit on the carbon source. So you can sort of imagine that depending on how the fungus enters this plant biomass, it has to do quite a lot of work to get into sugars that it can actually consume. Lignin is typically more a hindrance than a benefit because it has aromatic compounds, and if not only are they hard to degrade, but also when you actually degrade lignin, you get monomeric aromatics, and for most fungi these are quite toxic. So this is a good way for the plant to defend itself, and actually also this whole structure is meant to ultimately make it hard for fungi to get there. Now, quite often people, when they talk about saprobic fungi, which will be a large part of my talk, people always say, yeah, but saprobic fungi, do they really deal with biomass? Because they can't really degrade biomass unless it's damaged. But of course, we should consider that biomass in nature is almost always damaged. You have branches that fall off the tree that cause physical damage. You have the wind hitting tomato plants and other plants that also causes damage. So actually, there is always going to be some damage on plants that fungi can use as access points. So even saprobic fungi are capable of degrading almost intact plants because there is always damage. Now, just to show you a little bit more the complexity of this, so here we see different polymers, polysaccharides, that are present in the plant cell wall. So we have things like cellulose, hemicellulose, pectins, and then we have storage compounds like starch, inulin, and gums. And if you just look at the cell wall polysaccharides, you can see that you need many different enzymes to degrade it. And the problem, of course, which is on one hand very nice, but it's also a problem, is enzymes have a tendency to be very specific. So they will cleave a particular sugar that's linked in a particular way to another sugar, and not the same sugar if it's linked differently, or a different sugar that is linked in the same way. So as a result of that, fungi typically need a large arsenal of enzymes. And most saprobic and plant pathogenic fungi that degrade plant biomass, in their genome we can find somewhere between 200 and 350 genes that encode these putative enzymes. Well, obviously, the fungus doesn't produce all these enzymes all the time, but it typically produces the ones that it needs. So what I'm going to talk to you about mostly in this talk is what we call conversion of plant biomass by fungi. So we typically don't talk about degradation, because while we might want to degrade the plant biomass to convert it to something what we find interesting, of course, for the fungus, it doesn't stop with degradation. The aim is to get these monosaccharides out of the polysaccharides, get them into the cell, get them into the metabolic pathways, and ultimately assist the fungus in its propagation and growth. So during this talk, I will address several aspects of this, some a bit more deeply, some in just glancing by, and hopefully that will show you that diversity is actually a big issue when it comes to plant biomass degradation. So I'll start with the extracellular enzymes, which is effectively the start of the process. They degrade the polysaccharide into monosaccharides, and there is a massive amount of them in fungal genomes. Now, so when we're thinking about diversity, often people think, yeah, you're talking about how many enzymes there are, but actually diversity is a much more complex aspect when you go to enzymes. Of course, you need to think about how do these enzymes link to their substrate? What do they actually do? Can they be classified based on amino acid sequence, which luckily they can, and I'll come back to that in a little bit. But just classifying that doesn't really give you the whole answer. So yes, we can do this, we can do the annotation, and luckily for most fungi in microcosm, you get these annotations automatically. But that is only part of the story, because the Casey annotation will give you hints, but it doesn't give you necessarily the full story. So I'll give you here a few examples, because it's always nice to work with examples. And I start with an example from the genus Penicillium. Now, Penicillium is quite well-known genus, particularly for production of penicillin, which was the first antibiotic that was particularly important during World War II. But also Penicillia are fungi that are typically quite good at degrading plant biomass. Now, normally what we see when we compare the potential of these fungi for degrading plant biomass, we typically see that their overall fairly similar in their gene contents. You can see that here for the top 8 species. You can see that, okay, here, for instance, for GH5, we go from 15 to 17 to 14. But then, interestingly enough, there are some species that for some strange reason are very different. And one of these species is a species we more or less discovered by coincidence, is Penicillium subpubescens. And what you can see here in this bottom panel is that for many of these families, we see quite a strong increase in gene numbers compared to the other Penicillia. And this particularly addresses families that are either related to hemicellulose degradation or to pectin degradation. Now then, of course, the question is, so why would a fungus have more enzymes? And I always like to think of these things as simple as you can, because ultimately all these things happen through evolution. And I think the basic core idea of evolution is that evolution remains if it's beneficial. If it's not beneficial, it will probably not maintain, it will disappear again. And then you could sort of think about these enzymes as a toolkit. And if you think about them as a toolkit, and you think, okay, so for instance, in the case of Penicillium subpubescens, we have 10 endoxylanases in these two families. If you compare that to the other ones, it's typically between 3 and 8. So why does it have more? Now, you could very simply compare this again to a toolkit. So if you go to a tool shop to buy some tools, you're not going to buy 10 identical screwdrivers. You will buy a set of screwdrivers, but each of them will do something slightly different. And we're sort of using that same hypothesis for fungal enzymes, that yes, they may all be endoxylanases, but probably they're not going to do exactly the same thing. They're not going to act on exactly the same part of the substrate. And because of that, it can be beneficial to have additional ones. Now, I want to show you that in the next couple of slides. So typically when we do these things, we like to do phylogenies, because phylogenies are very indicative, because it tells you a little bit how related are these enzymes. And for instance, could they be originating from recent gene duplication, or are these more ancestral type enzymes that already might have been around for a long time? And if they've been around for a long time, most likely they have a benefit. So you can see that here. So here you see two phylogenetic trees, one for family GH10, one for family GH11. And in larger font and in bold are the members from Penicillium subpubescens. Now, as you can see here in family 10, one of them is sitting here, one of them is sitting here, one of them is sitting here. So yes, they're all family 10, but not particularly closely related. If we then look in family 11, where we have even more enzymes, you can see that, okay, in some cases, they are relatively close to each other, but we still see other enzymes from other species in between. So these are really not very recent gene duplications. So there must be a reason why Penicillium subpubescens decided it's convenient to have more endoxylanases. So what we decided to do is to have a look at the product profiles of these enzymes if we incubate them with xylem. Now, what we already know from a long time ago is that GH10 endoxylanases have different product profiles from GH11, and you can sort of see that if you compare panel A to panel B. But that's actually not the part which is interesting, because most of the time in literature, people assume that if something is a GH10 endoxylanase, it will do the same as any other GH10 endoxylanase, and the same for GH11. Now, if that were true, then it makes no sense to have more of these enzymes. So we thought maybe this is not quite true. And actually, if you look very carefully, you will also see that. If you just look here at the product profile in GH10, we see these three different Penicillium subpubescens GH10 endoxylanases. And you can see that, for instance, here we see xylotriose produced by this enzyme, by xin-A, but not by the other two. And in this particular case, we see this additional oligosaccharide appearing also in xin-A, which is largely absent in xin-B and C. And if we then go to GH11, we see a very similar thing. So here we have D, E, F, and H, and I. And you can see again, if you look at the profiles in more detail, you can see, for instance, here this peak appearing in H and I that is absent in the other ones. But also at the same point, you can see that the balance between the peaks is different. So clearly these enzymes are not the same. And then you sort of have to wonder if this is beneficial to the fungus. It probably means that the structure of xylem might be much more complex than we always think. And actually, to be able to efficiently degrade xylem, you need these enzymes that can all cleave these very different specific linkages. Now, of course, this is not typical just for GH10. It also occurs in other ones. And a little while ago, we studied a family called GH30 because it was a bit of a funny family. In bacteria, it's been quite well studied, but in fungi, the studies were quite limited. And typically, people would refer to this family as an alternative endoxylenase, so an alternative to GH10 and GH11. So again, we did the phylogeny, and actually the phylogeny was quite beautiful in this particular family because we get different clades, as you can see here. In the fungi are only present in 4 clades, the other ones are bacterial clades. But if you look at these clades, you can actually see that they seem to separate based on specificity. And we confirmed that by looking at the activities, and we could actually see that the different clades act on different polysaccharides, where here we have enzymes acting on beta-glucan, here we have enzymes acting on galactan, here a different class of galactan, and here we have enzymes acting on xylan. So actually this is already quite nice. We're thinking, okay, so we have this big family of many different enzymes, but based on phylogeny, we can already predict which substrates they'll be acting on. The question, of course, then is, so if they act on the same substrate, does this also mean they actually do the same thing? So this is what we then tested, and we were quite surprised, because initially when we tested the group that was acting on xylan, you can see the 4 enzymes here, and you can see the top enzyme only produces xylobioes, which is quite unusual for endoxylenases, because endoxylenases typically make a range of products. This seems to be a very specific product, and we actually named this gene xylobiohydrolase because it's specific for that. Then the next 2 enzymes, they seem to be losing their specificity, because the next one starts producing a little bit of another oligosaccharide here, and then if you look at the next one, you can see a little bit here and a few more here. But then the 4th one is even more surprising, because the 4th one actually produces barely any xylobioes, but it produces this huge range of different oligosaccharides. So here again, we have 4 enzymes from the same family, actually from the same subgroup of the same family, with a completely different product profile. And if you then look at the phylogenies, then it gets a bit confusing, because this enzyme here, that is cleaving as a pure endoxylenase, okay, that is sitting a bit far away from the other ones. But if we're looking at these 2 that are sitting very close together, they're from the same species, you can actually see that one of them is xylobiospecific and the other one generates a range of different oligosaccharides. So in this case, it also shows that even sequence similarity and phylogeny cannot help you really predict the functionality of the enzyme. Now we're hoping, and we're moving into that now, I don't have data yet from that by using AlphaFold and structural analysis, we can better predict the functionality of these enzymes and to basically do maybe some AI-inspired gene synthesis to ultimately make the optimal enzymes. Now, so this is talking about specific enzymes, but as of course, as I said already before, most of these fungi, they have between 150 to 350 enzymes. So the question is also, so how do these fungi choose which enzymes to use and which not to use, and do they typically choose the same ones? So we studied this in the genus Aspergillus, which is one of our favorite group of fungi, to try to see if we could make some kind of comparison between what the enzyme or the fungus would need based on the structure of the substrate and what it's actually doing. So here what we did, we used the Casey annotations again. We compared 17 Aspergilli, and we used proteomics to see which enzymes were actually produced in each of these species. And this turned out to be quite surprising. So if we first look at the genome content, so these lines separate different sections of the genus Aspergillus, you can see that yes, at the genome level, there is some variation, but actually, especially within the sections, they seem to be quite conserved in the number of genes that are present in the genome. Now then, when you do proteomics, we get a whole different story. So here, to explain the figure a little bit, on the right-hand side, this bar here, that is sugar beet pulp. So it consists mainly of cellulose and pectin. What we then did for all the protein enzymes that were produced, we sum them up based on the substrate they act on. So if they're xylene acting, they get put together. If they're pectin acting, they get put together. And then based on that, you can basically see what is the preferred component of sugar beet pulp that each fungus acts on. And then what you can see is actually quite surprising. As I said before, this group of fungi is highly similar, this subgroup, in their genome content. But if you compare, for instance, this one here, Aspergillus tubigensis, to Aspergillus brasiliensis, then you can see that tubigensis is mainly producing pectinases, not really doing much with cellulose, whereas brasiliensis is doing exactly the opposite. And you see the same thing in other groups. Here, for instance, in the group Nederlandis, you see that Nederlands likes to degrade cellulose, whereas Sedawi and versicolor focus on pectin. So even though they have the same set of tools, they choose to use different ones. Now, of course, you can hypothesize and philosophize a lot about all this, but the most likely explanation for this is that most of these fungi, they do not live in isolation. They actually, in many environments, you will find these different species of Aspergillus living together. And of course, if they would all be chasing the same component, that means they would be at war all the time. Whereas if they would be focusing on different aspects of the substrate, they could actually be benefiting each other by having a combined hydrolysis pattern. Now, this is speculative. I don't think there's any real evidence yet that this is what's going on in nature, but we have done some mixed cultivations, and we do see that in some cases, mixed cultivations result in better hydrolysis than single cultivations. So that is quite possible, and we are starting to go into this field a little bit, what we're calling this evolution of communities. So that actually the evolution is not driving the development of the individual species, but is driving the community as a whole, which forces some species to go in one direction and other species to go in another direction. And probably if we would be able to mimic real life in the lab, maybe we could actually try to replicate this type of evolution of communities and see that we could actually build up very efficient communities. In the lab, as Isabel knows better than most, actually having stable co-cultures is quite a challenge. So I'm not so sure how quick we will be able to do this, but maybe also that's where AI will help us to try and understand some of these aspects. So I was talking about the extracellular enzymes, which is only the first step of the process. And I want to briefly go into the transport. I'm only going to have one slide for you on that because I don't have enough time to go to every aspect in complete detail. But I do want to talk about it a little bit because, interestingly enough, transport obviously is very important. If you don't take up your nutrients, of course, you're not going to live. But surprisingly, the studies in fungi, especially filamentous fungi, on sugar transport are very, very minimal. There is a lot of work done in Saccharomyces, which is typically on glucose transport or on hexose transport. But in filamentous fungi, there's very little work. So we started some time ago on this. We started looking at it mostly from a bioinformatic point of view, using the available data to try to predict some functions. And interestingly enough, when you then start comparing fungi, actually there's a massive difference. So what you see here is a phylogenetic tree of a subset of fungi. So here we have Ascomycota, here we have Basidiomycota, Mucoromycota. And then immediately what you can see is that the richness of sugar transporters in Ascomycota is way higher than in all the other ones. So surprisingly, the other fungi seem to be much more limited in a way in their uptake systems than the Ascomycota. Now, if you would make the same picture for the K-zymes and you would make the same picture for metabolic enzymes, you would actually get a very similar picture. So it looks like that in Ascomycota, the diversity within a species of the genes involved in carbon acquiring seems to have expanded massively compared to the other classes of fungi. Why, we don't know. It's just something that we've observed. Now, interesting, there's a couple of nice separations there and nice examples. And that's, for instance, if you look here in the Basidiomycota, you see that while they are overall quite reduced compared to Ascomycota, the one group where they're quite well present is the disaccharide transporters. And actually, one of the ideas which we currently have about Basidiomycota is that rather than releasing monosaccharides from biomass, they seem to prefer to release disaccharides. And that may be because that way they can compete better with the Ascomycota that typically grow faster, because Ascomycota are more focused on monosaccharides, and it actually allows them to acquire carbon rather than having it stolen from them by the Ascomyces. Again, very speculative, but so far all the data we have seems to support a focus on disaccharides. We also typically in Basidiomycetes, we see very few exoacting extracellular enzymes, whereas we see quite a lot of exoacting intracellular enzymes. So all of this seems to support a mechanism where the biomass is degraded to disaccharides before it's transported rather than to monosaccharides. As I said, I'm not going to go too much on this. There's a paper here that if you want, you can look at it in a bit more detail. Transport's very interesting topic, but we certainly need a lot more data to really make sense of what we see there. So then let's go to one of my favorite topics. I quite like metabolism, mostly because we have a tendency to think that we understand metabolism. And I'm going to try and show you in the next number of slides that our understanding is probably very limited, and we probably oversimplify metabolism to an extent that could explain why metabolic engineering in industry is quite often very unsuccessful. So metabolism is something that, of course, is crucial for the cell. It gets its sugars, but the sugars itself are not that useful. The sugars need to be converted into compounds that the fungus can work with. So the first thing what happens when it enters the cell is that basically these primary carbon metabolism pathways convert sugars into very core components. The nice thing is that very often these metabolic genes are co-regulated with the Casey genes, and that sort of makes sense because if you release the sugar, you want to also convert it and not leave it for someone else. Now, up to, I would say maybe 10 years ago, typically most of these metabolic pathways were considered to be single gene, single step pathways. So you have, for instance, you have a pathway that has 5 steps, so you expect that 5 enzymes are involved in the pathway. Now, what we have seen in recent years is that actually the complexity of these pathways seems to be much higher. Many steps are not covered by a single enzyme, but by multiple enzymes. And we also see that within fungi, we have seen diversity of how these pathways are organized. So I'm going to show you some examples of that. So to start with a map, so this is basically primary carbon metabolism in fungi. As you can see, it's quite a complex map. The green compounds are the compounds that can come from the biomass. So that's the input, let's say, into metabolism. And then we see all these pathways. Ultimately, more or less everything goes to pyruvate. And pyruvate can then go into many directions, can do the acetyl-CoA, and this is where all the cellular components are made from. But as you can see, the pathways to get there are quite diverse. So arabinose and xylose comes through the pentose metabolic pathway. They go through pentose phosphate pathway before they reach central metabolism. Fructose can go rather direct. For galactose, there are multiple pathways to get there. And also for galactonic acid and rhamnose. So if you look at this pathway, and we did this in quite a lot of detail in Aspergillus niger, we can find genes for almost all the steps. But then when we take this model and we use orthology to find these genes in other species, actually it's not so simple. It works quite well for related species like Aspergillus nigelens or Penicillium, but if you go further away, increasingly, as indicated by these circles here, we cannot find the obvious gene purely based on orthology. So actually the homology between metabolism at maybe the pathway level, but most likely also at the gene level, is not that high across the fungal kingdom. So one of the ways to solve this, and that's a project that's currently ongoing, is that together with a consortium of PIs, we are trying to make a reference model for every group of fungi in the fungal tree of life that would basically allow you to have a nearby reference. So we should be able to predict most of the genes in the species surrounding that reference gene. Not going to go into that now. What I'm going to go into a bit more is, so what is the effect of these pathways? So again, coming back to nature, where typically fungi grow on plant biomass that doesn't consist just of glucose or just of arabinose, but it typically consists of a number of different monomers. So how important are these pathways if you grow on a substrate that's heterogeneous in composition? So what we decided to do, because in Aspergillus you can do a lot of nice genome editing, we decided to block each pathway at a step that we know if we block that pathway, we see no growth. So we blocked the pentose catabolic pathway at the last step before it continues to the pentose phosphate pathway. We blocked glucose and fructose consumption at the first step. So basically, if you don't phosphorylate them, they basically don't go into glycolysis. We blocked mannose at the first step as well. Galactose, we had to block 2 pathways, this one and this one. We actually think that this pathway, that's why I'm ignoring it at the moment, that this pathway actually doesn't exist. I think that's the paper that suggests this pathway is probably slightly misinterpreted its results. We're working on that at the moment. And then here for galactonic acid, we blocked this pathway and we blocked this pathway. So what we did is we blocked all the pathways individually, And then we also made a strain in which we blocked all of the pathways together. And then we wanted to see what is the effect that this has. So first, of course, we tested the effect on the relevant monosaccharides. And then you can, for instance, see that for the hexokinase glucokinase mutant, there's no growth on hexoses. But there's still growth on all the other sugars because the other pathways are still there. If you knock out XKI, there's no growth from xylose and arabinose. The rhamnose pathway is blocked, gives no growth on rhamnose. Galactonic acid, same thing. And for galactose, if you block both pathways, there's no growth on galactose. So that confirms that the pathways are actually involved in the sugars that we are wanting. Then, of course, what you can see, if you block all of them, we lose growth on almost everything. But surprisingly, we get growth on galactose. Now, we don't really understand that still. Galactose is a very enigmatic carbon source for Aspergillus niger. So I'm not going to go into detail there. But the main thing to see is that actually the individual pathways seem to work. As I said, I don't understand if we block all pathways why we actually see increase of growth of galactose, but let's just ignore that for today's seminar. Now then, if you go away from these pure carbon sources to more complex carbon sources, and I just have glucose here as a reference. So of course, maltose, you see the same effect as with glucose because it's just a disaccharide, same with sucrose. But then when you start going into the more complex substrates, you see interesting patterns. So the xylose kinase mutant, we see no growth from xylane. It sort of makes sense as a polymer of xylose. In the case of pectin, we actually see a little bit of reduction on the galactonic acid pathway. But then surprisingly, when we knock out hexose utilization, we lose growth from pectin. We have no idea why. It makes no sense compared to this graph, but we see that it happens. Similarly, on starch, of course, like with maltose there, but then if we grow on wheat bran, we do see an effect when we block pentose catabolism, but we see a much stronger effect when we block out hexose catabolism, and it's more or less the same as when we block everything. So there are some surprising things and some things that were sort of expected. And then we're trying to think, okay, so how does this all work? So we went a little bit further and we looked what happened during growth on these substrates. So here we're using wheat bran, which consists mainly of xylose and arabinose, and here sugar beet pulp, which is mainly galactonic acid and arabinose. And then if you look during time, what is happening? So what the fungus needs to do, it needs to first degrade the polysaccharide, release the monosaccharide, and then it can consume the monosaccharide. And you actually see this very beautifully. If you look at the different strains, let's ignore the last two for the moment, you can see that typically you first get more or less a reduction of some glucose. Then we see here in wheat bran, we see that basically there is some arabinose released, there is some xylose released, and then later this is consumed again. In the case of sugar beet pulp, we see that also galactonic acid is released and then later consumed, except in some strains. But overall, what we do see is we see that basically glucose, especially in the last two strains, is not consumed. So quite interestingly, these consumption patterns do make sense. Sorry, my light is on. There we go. But it is quite complex because actually these mutants, even though in the cases like, for instance, here where we block the galactonic acid pathway, we don't see accumulation of galactonic acid. So it's clearly much more complex than just a matter of one pathway stops one component, everything else keeps going. All these pathways interact. And one of the things that we think might be a reason for that is that some of these pathways, they generate cofactors like NAD and NADP, whereas other ones use them again. So probably having all of them run simultaneously allows you a very good balance of cofactor use and cofactor generation. Whereas if you block some of these pathways, you're actually upsetting a system that has evolved to be highly efficient. Now, that is one aspect. So we can see that if we block pathways, clearly the fungus is quite clever. You block one pathway, it uses something else. You block all of them, it can still do a little bit, but not so good. Now then, of course, the problem comes. Could it also be that in some of those cases, maybe genes that are normally dormant become activated? So we think we deactivate the pathway, but maybe that's not the case. So we trying to study that. And as I already said, so we already found in earlier studies that in some cases, it's not one single enzyme that converts a certain step, but it can in some cases be even three. So this is what we did quite a while ago with one of my PhD students. We found there were three enzymes here, two here, two here, 3 here, 3 here, and only the last step, which was the one we blocked in the previous study, is a single enzyme step. Now, you might think, okay, so then we know. But interestingly enough, this is on xylose and arabinose. On xylose and arabinose, when we knock out these genes, we see no growth. But we still see pretty good growth on wheat bran. So we're sort of thinking, maybe there is a bit more. So we went back into the genome and said, okay, can we actually find paralogous genes to these genes that may be also involved? So one of my PhD students did this, and she found 5 additional genes here in blue in Aspergillus niger that are highly similar in a phylogeny to the 3 that we already had. Surprisingly though, and this is something you can see, so the green ones here, so this one, and this one, are highly expressed on xylose and arabinose, which are the 3rd and the 4th ring of the circle, whereas these new ones, this one, this one, this one, they are typically not very well expressed on xylose and arabinose. So that could very easily explain why on xylose and arabinose these 5 genes have no function. Still, we wanted to see what do these things do. So the first thing we did is that we produced them in E. coli and we just tested what they in vitro can do. And the thing that they have in common is that all of them can convert xylose and arabinose. So even though we don't see expression on these 2 substrates, these 5 enzymes, so SLE-A to SLE-E, they are all capable of converting xylose and arabinose. In addition to that, actually there's quite a difference in specificity for all kinds of other sugars, but this seems to be the thing they have in common. So then we thought, okay, so if they can convert xylose and arabinose in vitro, and we don't see expression in vivo, is it then purely the expression pattern that prevents them from being involved in xylose and arabinose conversion, or is there more going on? So what we decided to do, and you see that in this thing, is that we decided to try to activate these enzymes. So here you see again, just for your reference, that if you compare the wild type to single mutants of the initial 3 genes, there's not really much of a phenotype. If you knock them all out, then we have no growth. However, if you knock only 2 of them out, so here we knock out RAA and XRB, it grows normally, XRA, XRB as well. Only when we knock out LAR-A and XRA, then there is a bit of a reduction. So XRB by itself cannot fully complement the loss of these 2. So then what we did, we took each of those genes and we inserted them in the location of one of the other 3 genes. So here we have these 2 genes knocked out, and then we replaced XRA by each of those 5 genes. And as you can see, no problem. Each of those 5 genes can fully replace the function of XRA. Similarly, each of them can fully replace the function of LAR-A. And then surprisingly, when we put them in the XRB locus, 3 of them actually perform better than XRB. So clearly we have 5 genes here that encode enzymes that are fully capable to support growth on xylose and arabinose, but for some reason, under normal conditions, on xylose and arabinose, we don't see this. Now, one of the things, of course, we're looking into now, so could it be that in some of the mutants, when we don't grow on xylose or arabinose, but we grow on crude substrates where there's different inducers, different conditions, or maybe under stress conditions, it could still be that these genes become activated, and actually they are in vivo playing a role in xylose and arabinose conversion, just not under the lab conditions that we initially tested. So, as I already said, we went from 1 gene for a step to 3 genes, and now we're at 8. And actually, since then, we've found 3 more. So basically, we now have 11 genes that are capable of converting xylose and arabinose to their respective polyols. And we actually don't understand why does Aspergillus need 11 genes for 1 step. Clearly, there is some reason for it. That's still something we would like to figure out. And to make it maybe even more complex, in some other Aspergilli, there's not 11, but there's 16. Let's see. So, but then of course we thought, okay, so there's all kinds of stuff going on. We also see that some of these enzymes, as you saw here, you see that they're active on some of these other funds, even on the other chiral counterparts. So maybe we don't fully understand the pentose catabolic pathway. Maybe we need to understand it a bit better. So this was the pathway we now had, where arabinose goes to ribitol, to alkxylose, and then to xylitol, xylose directly, and then xylitol goes to deoxylose and then to deoxylose 5-phosphate. And these are the genes that we knew were involved. So then we thought maybe we should look a little bit closer at these metabolic mutants. So we did the growth on the usual intermediates, and we see the phenotypes that you would expect. For instance, xylose doesn't grow on any of the mutants because it has to go through this one, through that one, and through that one. So if you block any of these, xylose cannot be consumed. In contrast, if you look at deoxylulose, you can see that actually it can grow quite happily still on this mutant because it actually doesn't need these genes to go to deoxylulose 5-phosphate. So then what about all these other sugars? So we tried L-xylose, and actually L-xylose grows quite happily on this mutant. Oh, sorry, it doesn't grow on this mutant. So it means that L-xylose somehow needs to go through xylitol. Now, L-xylose is considered a non-natural sugar. It's considered not to be present. But then why does Aspergillus is able to convert it? So maybe it is sometimes there. Same with D-arabinose and D-arabitol. We don't see a phenotype now on these mutants. So apparently they don't go through the same pathway as L-arabinose, but they go somewhere else. So what we postulated then in this figure is that actually this is the pentose-catabolic pathway for arabinose and xylose, where L-xylose can also go in, possibly through LAR-A based on the biochemical analysis, and also goes to xylitol and then basically goes this way. Whereas D-arabinose and D-arabitol don't actually go in through this way, but they actually come into the pathway at deoxylulose. And we have no idea at this point what genes are involved in that. But it basically shows that Aspergillus hasn't just a pathway for its, let's say, commonly found sugars in nature, but it has also accommodated the uncommonly found sugars to be able to use any carbon that it might encounter. Now, when you then do the transcriptome analysis to look at induction, we can quite easily see that induction of the pathway of the regulators RR and XNR is really very specific. We don't see it on most of the compounds, and if we summarize this, what you can actually see in the case of RR, we see very strong gene expression on both arabinose and arabitol, making us conclude that it's actually arabitol that's the real inducer rather than arabinose. Whereas in the case of XLNR, we only see strong induction on xylose and not on xylitol, and also not on L-xylose, which also goes through xylitol, suggesting that actually xylose itself is the inducer. So by using a combination of regulatory mutants, metabolic mutants, you can really dissect these pathways, not just for the genes that are involved, but also for the compounds that trigger expression. Now, just to jump out a little bit, and I know this is an evolutionary group, but I do think in the end we need to understand that what we do in science should amount to something in society. So I just want to take you, why is this particularly interesting to look at the pentose-catabolic pathway? And the reason for that is that actually xylitol, which is one of the intermediates, is a highly interesting product from an industrial point of view. It's used in chewing gums, as most of you probably know, in toothpaste. It's a very good alternative sugar. The problem is producing it chemically is not particularly nice. So to produce it chemically, you need high temperature, high pressure, typically nasty solvents. So you don't really want to do that. What is also being done at the moment is that people make this xylose hydrolysate, and they convert that by, for instance, Saccharomyces or by a bacteria. But producing this hydrolysate is actually also quite expensive because you need all these enzymes that are involved in that, and enzymes are expensive. So when we realized that we can actually engineer this pathway relatively efficiently, we thought, let's try something. Let's see, can we engineer Aspergillus that we take a crude substrate, we give it Aspergillus, and we get xylitol. So we tried to do this, and we thought, okay, we have several options. So what we did here is basically, is that we figured we want xylitol. So what we don't want is that this gets converted. So we have two options. We either block it here, or we block it at the next step. So we did that exactly. So we made a mutant that is knocked out here. We made a mutant that knocks out everything here. And then we made an additional one that also prevents the conversion of xylitol back to L-xylulose. And then we thought, okay, so let's have a look. If we make those mutants, can we accumulate xylitol? Now, the nice thing is maybe to remember for most of you, because maybe most of you haven't heard of this, but actually fungi do not like to accumulate things in the cell unless they need it. So what they typically do, if they accumulate something because of your actions, they will throw it out. So the nice thing is that if it starts accumulating xylitol in the cell, it will actually secrete it and it will end up in the broth, which makes it much easier to first of all identify, but also to extract. So we did this for these strains on these carbon sources at different time points, and then we had a look. And then, quite interesting, what do we see? So in the case of low xylose, what we see is that we see some accumulation of xylitol in the mutant that has XKI knocked out, in the triple mutant, as well as the one that has this one. And the levels are quite similar, although XKI goes down a little bit. If we then go to a higher xylose, so 1%, 2%, then clearly the XKI mutant is the winner. It produces the most xylitol, and it even continues to quite a late time point. The only problem is xylose is also expensive. So maybe that's not what you want to do. If we use xylane, you sort of, when you grow on xylane, you're sort of at low levels of xylose, and you can see that actually the patterns between xylane and low xylose are quite similar. But if you grow on wheat bran, then something very funny happens. All of a sudden, this mutant that's doing so well on pure xylose, for some reason, it only produces xylitol for a very short time, and then on wheat bran, something is induced that just takes the xylitol away. So apparently blocking it at the late step doesn't work. However, if we block it at the other step, we can pretty much continue production until 55 hours before the fungus figures out some way of converting it again. So it basically shows that it's not just about blocking a step, it's about blocking the right step in relation to the right substrate. So obviously this is not yet at levels that we were very happy with. Also, we basically felt it took quite a long time. So we tried to think, can we improve that? So here we have just a metabolic mutant. And then we said, so what if we start messing around with the regulatory mutants? So one of the mutants that's quite interesting is a mutant called CREA, because CREA can reduce expression if there's high amounts of monosaccharide. And maybe when we're accumulating xylitol, CREA gets activated. So we compared this triple mutant that is making xylitol to the triple mutant in which we also knock out this system. And actually what we see, we get a lower productivity of the strain than we have when we have a wild type. So clearly this is not by itself a good idea. So then we thought, so what if we over-activate the activator of the pathway? Theoretically, that should also work better. So again, what we did here, we made a constitutive version of the activator, and then what we see, we get a little bit earlier production of xylitol compared to this strain, but for some reason we also lose it very quickly again. So also that's not working. But surprisingly, when we combine these two, so even though this one delays production and that one speeds up production, if you combine the two, production speeds up even more and goes to higher levels, and we reach the highest productivity that we've had ever. So it just shows how complex it is to make a fungal cell factory. You need to manipulate the metabolism, you need to manipulate the regulatory enzymes, and we haven't even started on the transport yet. So it's actually not surprising that there are not that many success cases of this type of metabolic engineering, because you really need to understand metabolism and then make the right choices. So as I said, I would end with a little bit of evolution and pathogens. And this is a study which we've only started a little while ago. And we're actually now in the process of writing the paper. And there's some very interesting things there. So the question we asked ourselves here, so if we have plant pathogenic fungi that grow on different host plants, and these different host plants have different cell walls, would evolution not drive these fungi to have similar Casey sets if they grow on the same host plant? Or maybe that's just not how it works, and maybe in evolution there are different ways to achieve the same thing. So what we basically did is to try to understand this, and we decided to go for the two groups of plant pathogenic fungi that have the most members, which are the dotidiomycetes and the sorariomycetes. Now these two groups are quite distant taxonomically, so the idea would be, can we see parallel evolution in dotidiomycetes and sorariomycetes if they are faced with the same host plant? Now I have to make one disclaimer up front. Actually, figuring out the host plant of a plant pathogen is not so easy, because people don't necessarily describe what it's not a pathogen of. So people find it on a certain plant, they say, oh, it's a wheat pathogen. Does that mean it's not a barley pathogen? We don't know. Maybe people never studied this one on barley. Also, sometimes the limitation is climate-based. So for instance, I know of a study many, many years ago, actually an application, where people found that in Thailand they had lots of trouble with Macropora oryzae on rice. It was killing a lot of rice. But they were talking to people from Western Europe who said, oh no, we never see Macropora on barley. So they started growing barley in Thailand, and Magna Porter killed it as much as it killed the rice. So it turned out that it was not the host plant that was limiting, it's just Magna Porter didn't really like Western Europe. So it's very difficult to be certain about the host plant range of a plant pathogen. So I have to put that as a disclaimer because that obviously affects our results. So what we did, we compared the KZ genes of 56 dotidiomycetes and 42 sorariomycetes. We only took KZ families that were related to a specific plant polysaccharide. Because some of these KZ families have enzymes that act on multiple things, like I mentioned for GH30. And of course, that doesn't help us in understanding the role there. So we had to do a bit of a filtering. And then what we did, we looked at the different families and the overall content. And actually then it was quite interesting. So if you do a PCA analysis on this data set, what you see that it seems to be getting a bit mixed. So don't look too much at the colors because we're still not 100% sure if that's all correct. But if you just look at the triangles versus the circles, you can see there's a big group of sorariomycetes here. There's a big group dotidiomycetes here, but there's also plenty of them that are mixed in with each other. So clearly we cannot say that the dotidiomycetes have a different K-zone from the sorariomycetes because it's not quite that simple. There's a different figure that shows it even more clearly, but trust me, on the screen that goes nowhere, so that you'll have to wait for the paper. Then we went a little bit deeper and we basically did a bioinformatic analysis where we compared all the things we could compare. So here you see a heat map of the number of genes per family. Families are down here. The colors here indicate to which polysaccharide they're linked to. And then here we have 4 different features. So the first one here is the class. So the green ones are the sorariomycetes and the pale blue ones are the dotidiomycetes. And already you can see they do not separate. Then the next column here is the lifestyle. We have a couple of them in there which are potentially endophytes, maybe endophytes or pathogens. But again, is it something we're probably going to remove from the paper because I was talking to several plant pathogenic people and they said, well, you know, how do we know that something is an endophyte? Maybe it's an endophyte waiting to become a pathogen. Or maybe the conditions under which you look at it are just not when it shows its pathogenetic side. And vice versa, maybe a pathogen can be an endophyte for a long time before it starts killing the plant. So endophytes and pathogens, not so sure. It gets interesting when you go to the next one. So this is the non-wood substrates and we compare dicots versus monocots. Now there's a very important thing to realize in dicots versus monocots. Monocots typically have mainly xylem and cellulose, whereas dicots typically have xyloglucose and pectin. So that, of course, has a huge impact on the enzymes you need to degrade. The last one is the wood ones. It's a little bit more tricky. So here, but what you can see here is that we actually see that some of the sorariomycetes and dotidiomycetes sit quite close together. You can see here one sorariomycete in the middle of a group of dotidiomycetes. Here we see them also a bit mixed. So clearly there is something going on there that is not based on taxonomy. So clearly there is some evolution happening that is basically pushing these ones towards the composition of its substrate. And this is even a bit more clear here. And this is actually, I think, unfortunately, this is the only really clear example we have because it's very, as I said, because of the host rate, it's difficult to interpret. But this group is just, I would say, almost too good to be true. So here you see a group of 5 fungi. 3 of them are sorariomycetes, 2 of them are dotidiomycetes. All of them infect monocots. And all of them have lost the majority of their pectinases. So this is a very clear adaptation to the composition of the cell wall based on what they actually need to be able to be a pathogen for this plant. And you see that it's more or less the same effect in both dotidiomycetes and sorariomycetes. So I would say this is a clear example of parallel evolution. Also, it clearly shows that apparently plant cell wall degrading enzymes are somehow involved in the pathogenicity process. Now, a little bit surprising here at the bottom, we see another group of monocot-specific pathogens, but they don't seem to have any cellulase or xylanases left. So they seem to not really rely on these enzymes. But we can say based on this is that indeed there is some correlation. We don't have enough data probably at this point. Also, as I of course said, genome content is one thing. Production of enzymes is a whole different story. We don't have proteomics data. We don't have transcriptomics data. That's something maybe for the future. But what we clearly can see is there is something going on. Now, if you then go a little bit deeper and you look at the individual enzymes, and these are typically all enzymes, enzyme families involved in pectin degradation, then what you can see is that all the pathogens that prefer dicots, they have an increased number of genes in these families compared to the ones that use monocots. And as I already mentioned, pectin is typically a dicot polysaccharide. So you can see here across 8 different enzyme families, we see the same pattern of enrichment of these enzymes in dicot-specific plant pathogens. So I do think we have a pattern here, but of course we need a lot more data to really make sense of it. So as I said, some indications for parallel evolution, clustering of species from both groups with a similar host plant. We see similar trends in specific enzyme families, especially pectin acting, but also we see some taxonomic trends. In some cases, we do see that taxonomically close species, even if they have different host plants, they have a similar casey pattern. So maybe they haven't specialized yet. Maybe they're still at the early stages of evolution. Or again, maybe their host plant range is much broader than we give them credit for. So certainly, we cannot say more enzymes is always better, because sometimes species with a small enzyme set seem to be just as good. But there's many, many things that should be studied here to really get the patterns clear. Now, with that, I want to finish. I think maybe I took a little bit more time than I should have, but I hope I still managed to keep your attention through the whole thing. So I think I sort of showed you that the approach for plant biomass conversion is diverse, even between related species, like for the Aspergilli. We see that diversity occurs at all levels. It's not just diversity at enzymes, it's metabolism, it's at transport, it's at regulation. And we see some indication for co-evolution. We have much higher complexity in metabolism than we previously assumed, and therefore I think in some cases we need to go back to the drawing board and actually rethink if metabolism is really going as we have been assuming for the last couple of decades. And I think one of the ways to achieve this is to actually go more and more into sequencing whole genera. So we did this for Aspergillus, we're working on Penicillium and Trichoderma. And actually having a whole genus sequenced, it allows you to see much smaller differences than if you just have a few species per genus. So having more data, I mean, I'm not saying that you always need more data, but certainly if you want to go towards AI, you want to go to machine learning driven things, there is no limit to how much data is better. More data is always better because the noise will disappear if you just add enough good quality data. But quality is the second thing. You cannot compromise on quality. With that, there's a lot of people to answer. So of course, a lot of this was done in my group, but we worked quite a lot with Adrian, especially, and Marcos when he was still there in Concordia. Also people from Wageningen University, people in Finland. And of course, very much the Joint Genome Institute and the Casey Group, which are of course crucial to do any of this kind of stuff. And with that, I would like to thank you for your attention.
Thank you. Thank you very much, Ronald, for this very nice talk and also illustrating the complexity of all this enzyme that are secreted by fungi. So is there any question from the audience?
I stop sharing?
You can stop sharing or not. Then we see everybody perfectly.
I can always come back if necessary.
Absolutely. So is there any question in the audience? And if not, we have one in the chat box already from Gopal, who is asking, are you looking at protein complexes involving these enzymes and substrate utilization?
Well, that's actually a more complicated question than you might think. So when it comes to the extracellular enzymes, in fungi, at least in aerobic fungi, that doesn't seem to be real complexes. So there are in bacteria like the cellulosome and in some of the anaerobic fungi. Aerobic fungi do not seem to make enzyme complexes, but of course you do need mixtures of enzymes to achieve efficient hydrolysis. When it comes to metabolism, that's again one of those things that has been hinted upon, especially 20, 30, maybe 40 years ago, that some of these metabolic enzymes actually form metabolic complexes where the multiple enzymes of the different pathways are actually connected and do all the steps sequentially while it's connected to the complex. Now, I haven't seen too much confident data about that. And now, of course, I also have to wonder, if that is true, then how do we accommodate all these paralogous enzymes? So it would be very interesting and certainly worth studying, but I think this would probably take about 5 or 6 PhD students to actually get an idea if these are exceptions or if this is a common situation.
Well, that's you're starting a new group, right? So
Yeah, but even then, I don't think I have immediately enough people to really attack that topic.
Okay, Matthew, please.
Yeah, quick question about the substrate activity profiles of the different enzymes. I'm just wondering how much that profile of activity might change depending on whether or not you've hit the optimum pH or buffer conditions. And could you see
it changes a bit? It changes a bit. So what we try to do, we try to stay as much in physiological conditions as we can. Now that's a little bit tricky, especially when it comes to Aspergillus niger, because I don't think it does it as extremely in nature, since nature has probably more buffering capacity. But in a liquid culture of A. niger, it can go from pH 6 to pH 2.7 overnight, which would have quite a strong effect on the enzyme activity. So we do try to buffer this a bit, but you're of course right. The thing is, of course, once you start thinking about that, then it gets really complicated, because then you have to also think about water activity. You might have to think about salt concentrations. You might have to think about other potentially influential compounds that could be there. So I think again, you get into these situations that it's, I would say, pretty much impossible to mimic nature. So the best thing to do is to go under standard conditions, and then at least under standard conditions you can say, okay, under these conditions we see these differences. And then, especially if we can get a little bit deeper into AlphaFold and structure-function relationships, maybe at a certain point you could link studies where they look at, let's say, the structural properties of an enzyme in different pHs versus the activity in different pHs, and you could maybe link the two together, where you could see maybe that a binding site opens up more or closes more depending on pH, and that affects specificity. But that I would say that the technology is there, but it's rather tedious work that is actually not so easy to encourage people to do anymore these days.
Any other questions? So I was wondering, Ronald, you presented all this complexity in the enzyme sets of the fungi, and I know for a fact that you're a specialist also on the regulation. So we have a lot of different enzymes that seems acting, in different contexts. What about the regulators? Because there are kind of few known and in general, right?
I didn't go into it because then we would have been here for 2 hours. So because actually I love regulation, it's my favorite topic. So
exactly. But if we take the example of the the very early one on the penicillium, these GH10 and 11 that you mentioned, is there anything you've seen on how they are regulated?
Yeah, so we have done some regulatory mutants in this strain, and we do see that they are differently regulated. Again, of course, we have to keep in mind that this is under lab conditions. Yeah. Which is not necessary. But so what we are seeing now, we have some evidence now that in addition to let's say the primary regulators that are becoming quite well known, I always had the feeling that there was a second level of regulation, that basically in addition to what I would call regulators, you have modulators, where the regulator activates the whole spectrum of genes, and then the modulators determine this one goes higher and this one goes low. And we actually now have some candidates, modulators, which we are currently studying, which hopefully we can use as an example to show that indeed regulation also is not a single binding activator or repressor, but it's actually a combination of primary regulators with modulators that ultimately determine this. And that will also explain why when a fungus grows, for instance, on a certain substrate, it can switch its enzyme profile over time to keep matching the remaining structures that are still there when some of it has already been eaten. So I think regulation is unfortunately much more complex. It depends a bit on the fungus. Again, there Aspergillus has both potential as well as complications, because Aspergillus has a particularly high number of regulators, has about 600 transcriptional regulators in its genome, whereas I think Trichoderma has about 250. So clearly, just like with metabolism and with the cases and with the transport, it seems that Aspergillus and Penicillium somehow went a bit overboard in fine-tuning everything. of course means that if you can figure it out in Aspergillus, I guess it would not be too hard to figure it out in other fungi as well, especially because Aspergillus is a bit easier to handle. But yeah, but regulation is tough, and I think in the end, I don't necessarily see that we ever will understand regulation in nature, because I actually don't see how we can mimic nature well enough in the lab, especially for regulation. What I do see we could do is if we understand the regulatory system enough in the lab, we might be able to use it to make really superior cell factories.
Mhm.
And I think maybe for some of these studies, we just have to accept that maybe nature's understanding is just a little bit out of our reach, but we can actually at least manipulate nature enough to make it work really well and contribute to a sustainable future.
Very good. Okay, last question, I think, because of time constraint. Gopal again is asking on repathogenic fungi. How about infection strategies versus gene enrichment?
Yeah, well, that's of course a huge issue because infection strategies differ a lot. And I'm coming from more a saprobic field, and I've collaborated with many pathogen people, and actually convincing pathogen people that, especially plant pathogen people, that there's other things that are interesting than effectors is not always easy. And certainly trying to convince them, especially when they're working on species that make an aprosorium, to convince them that enzymes might have a role is also not easy because they think it's all mechanical. Luckily, not too long ago, we published together with people in France, we published a paper that shows that even during aprosorium formation, a limited set of KZ genes is expressed. And they actually showed that these enzymes that are produced are most likely just weakening the cell wall enough that the turgor you need in the aprosorium actually doesn't need to be as high as otherwise would be the case. So I think there is now some evidence that irrespective of the infection strategy, the enzymes do contribute. To which extent, of course, depends on the species, depends on the host plant, etc. But I would say that there is starting to be a realization that also infection process is not just about infection, because also you could argue that if you can get in the plant, but you can't degrade the plant and get your food, then it basically doesn't matter much that you get in. So ultimately, once they get in, they still need to degrade the cell wall to get the carbon sources. So I think we cannot ignore the fact that degradation has a role, but it might depend on the species at which stage of the process.
Well, that's a very good conclusion, I think, for bridging different specialties in our field. And with that, I think I will handle the seminar, at least for the question, with everybody except the professors. To, I believe, Penelope. Penny, are you here? Yes. Hi, Penny. So I think I already made you co-host, so I'm going to stop recording now. And